So we'll move on next to our next speaker, Dr. Anneta Zintl from the veterinary school in UCD. Anneta is a zoology graduate from Trinity College, where she also completed her PhD. She worked as a Newman Fellow in the veterinary sciences after as a postdoc, and she took up a lectureship in 2012 in the School of Veterinary Medicine, where she lectures on veterinary immunology and parasitology to veterinary medicine and veterinary nursing students. Her research interests focus on the epidemiology, detection, transmission, and control of various parasites, with the chief aim of investigating their importance for animal and or human health in Ireland. Her long-term research interests are common and emerging parasites of livestock and ticks and tick-borne diseases, and she's one of the national authorities on tick-borne diseases. I think in the context of climate change and changes in farming practices, these vector-borne diseases are assuming a greater importance and may be found in areas which they may not have been seen before. So I think this is a timely presentation from Anneta to talk about the role of tick-borne diseases in Ireland, in both people and animals in Ireland. So over to you, Anneta.
Thank you very much, Damien. Can you hear me all right?
We're hearing you loud and clear.
Great. Yeah, thank you very much for the invitation and for the very kind introduction. So I've been working on ticks and tick-borne diseases for a couple of years, and I'm just really going to give a very short overview of some of the work that we've been doing. So what I want to do is just give an introduction into tick distribution and the biology, and into the pathogens that are carried by ticks in Ireland and their zoonotic potential. And then I'll try to assess the current zoonotic threat associated with ticks in Ireland. So very briefly, the most important tick by far as vectors and vector-borne disease is concerned is Ixodes vicinus, or the castor bean or sheep tick. And it is widespread throughout Europe and throughout Ireland. So it has very strict habitat requirements. So it really needs a relative humidity of around 80% or higher. So as a result, it is usually found in places where there's good vegetation cover, particularly in the thick leaf litter under the vegetation. And it becomes active at temperatures between 7 and 10 degrees Celsius. So mostly in continental Europe, it is mostly found in woodlands, but in places where there is a lot of precipitation, it can also be found in open ground, in hedgerows and rough vegetation. And as you can see here in the map of Ireland, we plotted the probability of ticks occurring in the various places in Ireland. As you can see, the places or the areas that are particularly suitable for ticks are the West Coast and the Shannon catchment. So Ixodes vicinus, in spite of the name the sheep tick, it actually has a very broad host range. So it can really infest all mammals, birds, and even reptiles. But it really likes to feed on deer, and wherever there are deer, there tend to be quite a lot of ticks, if the other environmental parameters are suitable, of course. So there are three stages in the life cycle of ticks: the larvae, the nymphs, and the adults. And all life cycle stages are obligate bloodsuckers. So they only ever feed on blood. And in fact, each of these life cycle stages only feeds once. And that's quite remarkable because the tick actually lives for between 2 to 5 years. So in this time, it only feeds 3 times. In between the feeds, the tick spends a lot of time in the environment where it digests the blood meal and molts to the next stage. And our ticks, Ixodes vicinus, they find new hosts by ambushing. So what they do is they crawl or climb to the tips of vegetation and wave their little front legs in the air and wait for a host to pass. And when there is someone coming by, they hold on with their front legs. So they can't jump and they can't fly. So now I'm just going to give a brief run through the pathogens that we know are carried by Ixodes vicinus in Ireland. So the first one here is Laupingel virus, which is of course an important disease in sheep. Now, a lot of the time infections go unnoticed, but sometimes the virus can invade the central nervous system, causing depression, loss of coordination and paralysis, and can be fatal in those cases. We don't really know how common it is in sheep. Damon actually did a study of the seroprevalence of Laupingel virus in sheep in Donegal about 10 years ago. And his team found that there was evidence of a lot of exposure to the virus, but only a relatively small proportion of sheep actually got sick with it. Now, humans can become infected with Laupingel virus. Here it can cause a flu-like disease, and sometimes there are neurological signs as well. But we actually don't know that much about the disease that it causes in humans because it's extremely rare. So, so far, no cases have ever been reported in Ireland, only 4 in Northern Ireland, and I think in the whole of the UK, less than 50. Also, interestingly, Laupingel virus in humans seems to be transmitted through contact with infected carcasses rather than ticks. The next pathogen, tick-borne pathogen I want to briefly mention is Anaplasma phagocytophylum. So it's a bacterium that invades the neutrophils, white blood cells, thereby causing an immune suppression. Now, the story about Anaplasma phagocytophylum is a little bit complicated because around 20 years ago, Dumla and colleagues, they combined the pathogens that infect horses, dogs, humans, and ruminants into a single species and called it Anaplasma phagocytophylum. And they did this on the basis of genetics. So these pathogens are genetically incredibly similar. But certainly, I think that the evidence that these are really distinct pathogens or certainly distinct strains is very strong. And as far as we can tell, the only Anaplasma phagocytophylum strain that occurs in Ireland is the one that causes tick-borne fever in ruminants. So far, there have been no cases of it reported in humans. And we also don't think there have been cases in horses or dogs. Colleagues of mine have been looking at dogs in the animal hospital as well. So as far as we can tell, then the only strain that's present in Ireland is the one that causes tick-borne fever in ruminants. So as the name would suggest, it causes a high fever, a loss of appetite, and because of this invasion of the neutrophils, it causes immunosuppression. So secondary infections are very common. And it's a particularly important disease, obviously, in young lambs and in pregnant ewes. We don't really know how common it is in Ireland, but we're currently carrying out a survey together with the regional veterinary labs and the blood testing laboratory in Cork to have a closer look at this. So as far as we know, it's not a zoonosis in Ireland. In contrast, this one here is Babesia divergens. It's a protozoan pathogen, a protozoan parasite that invades the red blood cells. And of course, it causes red water fever in cattle. And this is because the parasite destroys the red blood cells when it invades, and it causes widespread anemia. And in fact, the anemia can become so pronounced that organs and tissues become anoxic. And it can cause very high mortality rates in cattle unless they're treated rapidly and aggressively. Now, we did some work over the years. These were all questionnaire surveys, and they indicate that the incidence in cattle of red water fever has actually declined in the last couple of decades. Now, Babesia divergens is infectious to humans. In fact, in humans, it can cause a rapidly fulminating malaria-like disease. but it's extremely rare, and it only seems to occur in splenectomized people. And overall, since the first case was reported in the 1950s, only about 60 cases in the whole of Europe have been reported. 5 of these occurred in Ireland. And then, of course, there's Borrelia burgdorferi, and of course, Margaret already mentioned Lyme disease. So it is, of course, the tick-borne zoonosis that everyone thinks of when they're talking about tick-borne diseases in humans. So it causes non-specific flu-like symptoms, but more severe manifestations may affect the skin, the nervous system, the joints, or the heart, depending on the Borrelia burgdorferi strain that is involved. And up here is just an image of the erythema migrans, the very typical skin rash that is produced and is often associated with the bite of an infected tick, but not always associated with it. So there can be infections without the skin rash appearing. In Ireland, Lyme neuroborreliosis, is notifiable as in the rest of Europe. And since 2014, between 8 and 21 cases have been reported per annum to the Health Protection Surveillance Centre. And based on these figures, the Health Protection Surveillance Centre estimate that there is a crude incidence of 0.5 to 4 per 100,000 of population every year. So as it is a zoonosis, there's of course a wildlife hosts that are serve as reservoirs for this infection. The competent hosts are rodents, insectivores, and birds. Deer don't seem to be susceptible to Borrelia burgdorferi infections, so their complement system seems to be able to deal with infections quite effectively. So contrary to popular belief, deer do not contribute to tick infection rates with Borrelia in the environment, although they do contribute to tick numbers because, as I said, ticks really like to feed on deer. No, now I've got the same problem as Margaret, I think, because I just try to, no. Yes, here we are. Okay, sorry. So we carried out a couple of tick field surveys between 2016 and 2019. And what we did was that we visited several places all across the island, including woodlands and farmland. And we collected ticks using this standard blanket dragging method that is shown here in the slide. And we brought the ticks back to the lab, identified them, and screened them for a range of pathogens. And these here are just some of the results that we got for the presence of Borrelia burgdorferi in nymphal ticks that we collected during this time. So we found that the overall tick infection rate, so the number of ticks or percentage of ticks that were infected with Borrelia ranged between 4 and 14%. And the highest infection rate was actually in woodland ticks. But when we subtyped these Borrelia burgdorferi strains, then what we found was that most ticks actually were infected either with Borrelia wallaceiana or Borrelia burgdorferi sensu strictu. So Borrelia wallaceiana actually seems to be apathogenic. So as far as I know, it's only ever been described from a single human patient, and that person may well have been sick with something else as well. But Borrelia burgdorferi sensu strictu is associated with arthritis and neuroborreliosis. Some of the other ticks in farm and woodland were infected with Borrelia guarinii, again, which is associated with neuroborreliosis, whereas Borrelia afzellii, which is quite common in Eastern and Central Europe and associated with chronic skin conditions, was really quite rare in our ticks. So how would I assess the current zoonotic threat associated with tick-borne pathogens in Ireland? It seemed from our work on bovine babesiosis and bedwater fever that the number of habitats and the size of habitats that are suitable for ticks seem to have actually declined over the last couple of decades. And also when we look at our tick infection rates with Borrelia burgdorferi and also the strains of Borrelia burgdorferi that we detected in these ticks, they were very similar to the rates that were reported by colleagues, Professor Jeremy Gray and his team in the 1990s. So they seem to have remained really stable over this time period. Also, we did carry out some work on screening ticks for a range of tick-borne pathogens that occur in continental Europe, and we found no evidence that the Irish ticks were infected with any new or unexpected tick-borne pathogens, but I should say that this was very much a pilot study. So how would I assess the current zoonotic threat associated with tick-borne pathogens in Ireland? I think, if anything, it may have declined in recent decades. I certainly don't think it has increased since the work was done by Jeremy in the 1990s. So this really just leaves me to thank our funders. So MSc Animal Health have been very, very generous over the years, giving us money to have a number of students working on this, but also Chagas has been supporting this work, and VectorNet, who are a European collaboration and support surveillance work. And then, of course, all of the postgraduate and undergraduate students that have been roped in over the years to help with the tick collections. Thank you very much.
Very much, Anneta, for a very interesting talk. As a kind of a vector nerd, I found that very interesting, and I always found that ticks were one of those things that tended to come at you from the left field. So I